Hello guys, welcome to this YouTube channel. A good day to everyone watching this video. In this video, I'm going to discuss the answer key for the GATE Excel 2018 paper for the biochemistry section. So let's get started. So the first question is, to which one of the following classes of enzymes does chymotrypsin belong to? So this is a pretty simple question. So it's basically a hydrolase, which is option B. So I would strongly suggest everybody who is preparing for these competitive exams to look at the mechanism of action of chymotrypsin as well as lysozymes and even ribonuclease A. So these are pretty asked questions, especially with the mechanism of action. So if you can look at how or what amino acids are being targeted and which one is like the donor pair of electrons or the acceptor of electrons, so you will be able to understand the mechanism of action, and based on that you will be able to say whether it's an oxidoreductase or a hydrolase or a transferase or an isomerase. So here chymotrypsin is a hydrolase, and its enzymatic action is based on hydrolytic action. Moving on to the next question. So the substrate saturation profile of an enzyme that follows Michaelis-Menten kinetics is depicted in the following figure. What is the order of the reaction in the concentration range between 0.8 to 1.4 molar? So let's just look at that region here. So it's from 0.8 to 1.4 molar. So clearly here in this particular region, you can see that the velocity of the reaction remains almost constant despite increase in the substrate concentration. So this clearly means that this is a zero order reaction. If you know basic chemical kinetics from your 12th standard textbook, this is a very simple question to solve. So the answer here is option A, which is zero order kinetics. Moving on to the next question. Which of the following conformations of glucose is the most stable? So here it's again a very simple question to answer. The answer is chair conformation, wherein the glucose is highly stable with less delta or with more negative delta G value. Yes. Moving on to the next question. Which one of the following profiles represent the phenomenon of cooperativity? So as an example, I would say hemoglobin binding to oxygen. So if you look at the curve of how hemoglobin binds to oxygen, it's a cooperative mode of binding. And here, most of these cooperative mode of binding have a sigmoidal graph. So here, the correct option is option C. It's a sigmoidal graph. So the reason why there is cooperative binding is to ensure that hemoglobin also has high affinity for oxygen because it's the major carrier of oxygen in the blood, and at the same time also to be able to release oxygen at the sites of tissues when these cells actually require them for their basic respiration. So it should also have high affinity to be able to transport as well as to release the oxygen to the targeted tissues or cells. Yes. Moving on to the next question. Which one of the following amino acids is responsible for the intrinsic fluorescence of proteins? So this is a very simple question, and the fluorescence of the proteins mainly comes from the aromatic amino acids, and here tryptophan is the right answer, and that is mainly responsible for the intrinsic fluorescence of proteins. And we also use spectrophotometric based methods to quantify the concentration of protein based on the presence of these aromatic amino acids. Alright, moving on to the next question. So the glycosylation of the proteins occurs, and this is a very simple question that any basic biology student should be able to answer, and here the correct option is Golgi apparatus. So the ER-Golgi network plays a crucial role in the post-translational modifications of proteins, and especially when it comes to glycosylation or the addition of glycosyl groups to these proteins once they are being translated, then this mainly occurs in the Golgi apparatus of the cell. Alright, moving on to the next question. Which one of the following properties of the myeloma cells is used in the hybridoma technology to generate monoclonal antibody? This is again a very simple question if you know how hybridoma technology works for the production of monoclonal antibodies. So here it mainly works on the principle of the fusion of the B cell that produces the antibody and the myeloma cells, which are like cancerous B cells that keeps on dividing at an uncontrollable rate. So these myeloma cells are being engineered in such a way that they lack these enzymes called HGPRT which is hypoxanthine guanine phosphoribosyl transferase. The reason why they are engineered to lack HGPRT is that, so basically these cells are grown in a medium called the HAT medium, which is the hypoxanthine aminopterin transferrin medium. So basically, thymidine medium. So basically in this HAT medium, these amino acid precursors these nucleotide precursors are given so that these cells are able to synthesize nucleotide by the salvage pathway. So B cells which are producing the antibodies, they are basically short-lived. So basically they won't be able to produce so many, they won't be able to survive this particular HAT medium for a long time because their lifespan is short-lived. However, they do have this HGPRT enzyme for the salvage pathway synthesis of these nucleotides. However, these myeloma cells, being able to divide at an uncontrollable rate, they lack this HGPRT, and as a result, they won't be able to synthesize nucleotides through the salvage pathway. The de novo pathway of nucleotide synthesis is blocked using this aminopterin in the HAT medium. So basically, nucleotide synthesis is being totally abolished in these myeloma cells, and hence they die. Ultimately, the fused cells, which are the B cells and the myeloma cells in combination, they survive, and as a result, they continuously also produce these antibodies, and also they survive this HAT medium. So that is the reason why this particular technology is used with this particular principle, so that it ultimately results in the continuous production of these monoclonal antibodies. And the correct answer here is lack of hypoxanthine guanine phosphoribosyl transferase. That is the correct answer. That is, these myeloma cells will lack these. Moving on to the next question. The movement of protons through the F0-F1 ATPase during the mitochondrial respiration is required for, this is again, I would request everybody to look at how the electron transport chain works. This is very important, especially with complex 1, 2, 3, 4, and 5 is the ATPase. So here it is, the correct answer here is option B. That is changing the conformation of F0-F1 ATPase to expel the ATP. So it's like this triangle that you have to remember, wherein one side you get the ADP, the other side you get the phosphate group, and then they ultimately combine, or they go join together to form the ATP. So this is like this triad group that you need to remember. The structure is pretty important, especially the tunnel-like structure or the tube-like structure that connects these two subunits together. So that is also pretty quite regularly tested in these GATE papers. Moving on to the next question. The number of NADP plus molecules required to completely oxidize one molecule of glucose to CO2 through pentose phosphate pathways. So before we go on, we should first understand the complete reaction for the pentose phosphate pathway. So let me write it here. In pentose phosphate pathway, in the oxidative phase, you will have glucose 6-phosphate getting converted to 6-phosphogluconolactone, and this gets converted to 6-phosphogluconate, and this ultimately gets converted to ribulose 5-phosphate, which is involved in nucleotide synthesis, ribulose 5-phosphate. So here, this is the overall reaction, and here one NADP plus, here you should remember that the NADP is used and not NAD. So NADP plus gets converted to NADPH, and again in this step, NADP plus gets converted to NADPH, and in the overall reaction, one carbon dioxide is released. Okay. So here they have asked, you should be very careful with the question here. Here they have asked, completely oxidize one molecule of glucose, which means ultimately this has to result in the production of 6 CO2, because glucose has 6 carbons. So for one carbon atom, for one carbon dioxide, 2 molecules of NADP plus is consumed. Therefore, for 6 carbon dioxides, it has to be 6 into 2, which is 12. So the correct answer here is 12. So if you know this particular reaction and where NADP plus is being consumed to give NADPH, then this is a pretty easy question to solve. And you have to be very careful with the words here. Moving on to the next question. So the measurement of absorbance of a solution containing NaDH in a path length of 1 centimeter cubed at 340 nanometers shows the value of 0.31. The molar extinction coefficient of NaDH is 6200 molar inverse centimeter inverse. The concentration of NaDH in the solution is what micromolar. So again, this is a classic example of Beer-Lambert's law, which states that absorbance is equal to epsilon into C into L. So here absorbance is 0.31, epsilon is already given, which is 6200 molar inverse centimeter inverse. Concentration is something that we need to find, and the path length is 1 centimeter. So now concentration would be equal to 0.31 divided by 6200 molar. However, they have asked the answer in micromolar. So please kindly check what units are being asked. So that will give you 0.31 divided by 6200 into 10 power 6 micromolar. So this will be equal to 3100, 3100, correct. 31,000. Sorry, 31,000 divided by 6,200. So if you calculate this, this will be 50. Yes, it is actually 3,100,000. 3,100,000 divided by 6,200, which is equal to 50. So the correct answer here is 50 micromolar. So be careful with the exponents here. Yeah, so it is very easy to get distracted and then miss the zeros here. So it's very easy to make mistakes here. So here the correct answer here is 50 micromolar. So be careful with the units. Moving on to the next question. Among the reagents given below, which one of the following combination of reagents will not break the disulfide bonds in the immunoglobulin molecules? So clearly, reduced glutathione does this job. Dithioethritol will also break the disulfide bonds. Methionine doesn't do it, and similarly SDS doesn't do it. So SDS equally distributes the negative charge during protein denaturation. So the correct answer here is option A, which is R and S, because the question here asks which will not break the disulfide bond. So reduced glutathione, dithioethritol, beta-mercaptoethanol, these things will break the disulfide bonds. However, sodium rhodocyl sulfate, which is SDS, and methionine doesn't do this. So the correct answer here is R and S. Moving on to the next question. Match the protein elution condition given in group 1 with the appropriate chromatography matrices from group 2. So this is a pretty simple question if you know the basic molecular biology techniques that we use for protein separation. So I would first start with histidine, because it's the most common question that is being used, because if you have high concentration of histidine, the NI-NTA affinity chromatography column is used. So I would first match this, and then increasing concentration of sodium chloride, that is being separated using DEAE cefacryl, and then decreasing concentration of ammonium sulfate is separated is used in phenyl cephorose, and decreasing concentration of H+ is used in chromatofocusing. So here the correct option is answer A, and I would request everybody here to know how the principle of each of these. NI-NTA is pretty common, however where DEAE, cefacryl, and chromatofocusing and phenyl cephorose is being used and what's the principle behind it is something that you all have to know. And since this video I'm only discussing the answer key, I'm just keeping it short and then just helping you with how to quickly figure out the options for the given questions. So yes, so the correct answer here is option A. Moving on to the next question. Which one of the following is not a neurotransmitter? So this can be a pretty tricky question if you don't know which type of molecules does what. So adrenaline here is definitely a neurotransmitter. Glutamate, yes, many people get confused, but glutamate, yes, mostly involved in excitatory signals. Histamine is also a neurotransmitter, however histidine is not a neurotransmitter. So the correct answer here is histidine, because the question here asked is not a neurotransmitter, and that's pretty simple. Moving on to the next question. The type 2 hypersensitivity reaction is mainly mediated by, it's very easy to tick the option IgE, because we all know that IgE is mainly involved in hypersensitivity reactions. However, the correct answer is IgM. So first of all, there are four main types of hypersensitivity reactions. I would request everybody here to go through the basic immunology, immunology textbooks and concepts for hypersensitivity reactions, the different types of hypersensitivity reactions. And here the correct answer is IgM, which is mainly involved in the type 2 hypersensitivity reaction. Okay. Moving on to the next question. Which one of the following reaction mechanisms drives the conversion of low energy 3-phosphoglyceraldehyde to high energy 1,3-bis-phosphoglycerate? So here the correct option is option B, which is oxidation coupled with anhydride bond formation. So first of all, you get a glycerate, so 1,3-bis-phosphoglycerate. So there is an anhydride formation between the phosphate and the oxygen groups here. You have a PO-P bond there, and then this is also an oxidation reaction because it's getting converted from 3-phosphoglyceraldehyde to 1,3-bis-phosphoglycerate, which is also an oxidation reaction. So the correct answer here is oxidation coupled with anhydride bond formation, or more precise, phosphoanhydride bond formation. Moving on to the next question. A polymerase reaction is carried out for 10 cycles in a volume of 1 ml with 5 molecules of template DNA. Assuming that the efficiency of the reaction is 100%, the number of molecules of DNA present in 100 microliter at the end of the reaction is what? So this is a very simple question with the basic formula for PCR that you can use, and that is N equal to N naught into 2 power N, where N is the number of DNA molecules at the end of this N cycle. So this is the number of cycles. Here it is 10, and for every cycle there is doubling of the DNA template. This is the initial number of DNA molecules, and this is the final one. So just substituting all of these, N is equal to N naught, which is 5 molecules of DNA, into 2 power 10. So which is 5 into 1024, which is 5120 molecules of DNA. However, this is in 1 ml. So this is basically in 1 ml. They have asked the answer in 100 microliter. So you divide this divided by 10. So for 100 microliter is 5120 divided by 10, which is 512 molecules of DNA, which is present in 100 microliter at the end of the reaction. So the correct answer here is 512. So that's a pretty simple question. Moving on to the next question. So the secondary structure topology diagram of 400 amino acid long protein X is depicted in the given figure. The start and end amino acid residue numbers of each alpha helix are marked. The percentage of residues forming alpha helix is what? So first of all, these are the residues that form the alpha helix. So they have clearly mentioned it. So here, how do you do it? So here the number of alpha helix residues would be 70 minus 21 plus 1. The reason why I gave plus 1 is that the 21st amino acid is also included in the alpha helix formation. So that is why you need to have plus 1. If you do 70 minus 21, then you are basically counting from 22 to 70 and not from 21 to 70. So this is basic mathematics that we all know. The plus 249 minus 225 plus 1 plus, so this is basically the the numbering of the amino acids along the entire protein. So if you want to know the number of amino acid residues, you need to basically do the subtraction along with plus 1, because the first amino acid in the alpha helix is also involved in the alpha helix formation. Plus 324 minus 320 plus 1, the whole divided by 400, because this is a 400 amino acid long protein, into 100, because they have asked it in percentage, right? They've asked it in percentage. So if you do this, you will have 50 plus 25 plus 5 divided by 400 into 100. That will give you 80 by 4, which is going to be 20. So the correct answer here is 20%. This is a very simple question to solve if you know basically how to do this. Moving on to the next question. An enzyme follows Michaelis-Menten's kinetics with substrate S. The fraction of the maximum velocity Vmax will be observed with the substrate concentration S is equal to 4 kms. So first of all, Michaelis-Menten's kinetics is frequently asked in most of the GATE biology papers, including biotechnology, and here of course in life sciences biochemistry. So let's first write that. So V equal to Vmax into substrate concentration divided by Km plus substrate concentration. So here they've asked that, they've already given that the substrate concentration is 4 times Km. Okay, so just substituting that, we will have V equal to Vmax into 4 times Km divided by Km plus 4 times Km. So that will give you 4 by 5 times of Vmax. And they have asked the fraction in decimal. So that will be 0.8 Vmax. So the correct answer here is 0.8. It's a very simple Michaelis Menten kinetics. You know, if you get such a question, you are really lucky because they have not added extra complications through these inhibitors and stuff. So in that case, you have to do a lot of calculations to understand how it or what type of inhibitor it is and things like that. So this is a very straightforward question. If you get it, it's pretty easy to score. Moving on to the next question. The mass spectrum of benzoic acid will generate the fragment as a base peak, that is the fragment with 100% relative abundance of M by Z ratio at. So here basically this is a fact that you would need to know, that benzoic acid being a parent ion or the precursor ion, I would say I usually call it the parent ion, before break down or before being broken down into these product ions, the most stable product ion is the phenoxide ion because it is resonance stabilized. It is resonance stabilized. So phenoxide ion has a formula C6H5 plus. Okay, so being resonance stabilized, this will be the fragment with the as a base peak, that it will have the 100% relative abundance. By relative abundance, I mean that compared to the other fragments, compared to the other fragmented ions or the other product ions, this one will have the maximum intensity, and hence the abundance of this is regarded as 100%, and everything else, all the other peaks would be compared with respect to this. So that's how mass spectrum works for proteins. So here, if you do the calculation, the molecular mass here is 72 because carbon is 12, so 12 into 672 plus 5, which is 77, and here the charge is 1, so M by Z is going to be 77. So the correct answer here is 77. So if you can actually figure it out that the phenoxide ion is the one that is going to be the most stable with being resonance stabilized, then this question is pretty easy to solve. Moving on to the last question. Here, the standard free energy delta G naught values of reactions catalyzed by citrate lyase and citrate synthetase are minus 670 and minus 8192 calorie per mole respectively. So here it's a big reaction. They have given two big reactions. So the standard free energy of acetyl-CoA hydrolysis is what? This is a very, very simple thermodynamics question. So here they have asked what is the hydrolysis of acetyl-CoA. So you need to know first of all the reaction that you need to expect. So here acetyl-CoA hydrolysis would be acetyl-CoA plus water or H2O giving oxaloacetate plus CoA. Yes, sorry. It is going to be acetate plus CoA. This is the overall reaction. So acetyl-CoA plus water gives acetate plus CoA. So if this is going to be the overall reaction, what you have to do from these two equations is you just have to add. So when you add these two reactions or when you couple these two reactions, the citrate gets cancelled and the oxaloacetate also gets cancelled. So that leaves with the reactants to be acetyl-CoA plus water and the products to be acetate plus CoA. So because this is a hydrolysis, you basically expect this acetyl-CoA to get broken down into acetate plus coenzyme A. Alright, so based on this, how do you get the standard free energy of this overall reaction? You just have to add it. So delta G, G prime or G naught, which is basically denoting the standard free energy, delta G prime 3, let me call it 3, would be equal to delta G prime 1 plus delta G prime 2. So that will be minus 670 plus minus 8192. So if you add both these, you will get the answer to be minus 8862. So this is a very simple question. If you know that hydrolysis would basically break down the reactant into simpler products. So that is the key here, and then you just apply the basic thermodynamics here. Minus calorie per mole. Okay. So with this, I'm completing discussing the answer key for the GATE Excel 2018 paper for the biochemistry section. I hope you found this video interesting and also useful to you, and I hope that you use these tips and tricks to solve your upcoming GATE exam questions, and hope your preparation is also fruitful. Thank you and have a great day. Goodbye.